Or silent.
I'm good.
Follow me on Roblox, guys.
Okay, listen up, please. Pay attention. So, proteins are basically what makes us what we are. There's what makes your eyes a certain color, your skin a certain color, your hair a certain texture. It makes whether or not you will have a high metabolism or a low metabolism. It also actually has a big impact on your personality. As a matter of fact, with psychology, it seems like every year they find more and more that it's what makes you you is basically the molecules that are inside your brain and stuff. So most of those are proteins or something similar to it, and not only do they serve a big purpose in the structure of our body, for example, our skin and muscles and everything are made out of protein, but they serve a very big functional role in that they form enzymes and other things like hemoglobin, etc., that carry stuff and basically do all the work of the cells as well. And so consequently, they're very complicated molecules. They are typically on the order of 50,000 amino acids linked together. And each one of those 20 types of amino acids that they have to choose from has a different side group. And by doing that and putting those all together, you get these basically molecular machines that can manipulate other molecules. And that's how they end up doing such incredible things that they do in the cells. So they basically run around like little robots and stuff and make everything happen. As a matter of fact, the whole process of making new proteins is run by, guess what, enzymes that make proteins. And remember the DNA replicating, DNA polymerase, that's what made it split. There's a bunch of other enzymes that do all the bring in the, or link the chains together and stuff like that. So all of those are carried out by proteins, so they are crucial. And it is also, of course, like I said, what makes you different from other people is the differences in your proteins. So we know that that is dictated by DNA. That's what, if you have, if you're a twin, an actual identical twin, your DNA is pretty much identical to your twin, and therefore that's why you look and act and everything else is very similar between the two of you, regardless of your upbringing. So if you've ever looked into separated twin studies and stuff, they're very fascinating. Anyway, so the DNA it has the instructions for that protein synthesis. So we're going to look at how that actually ends up making a protein. The DNA itself stays in the nucleus. It's like a library, okay, or it's like maybe more like a vault. And I think the purpose is there to stay in there is to keep it protected. Yes, put your phones down, please. But to keep that original copy protected. And therefore you can copy it and you can make sure the next cells, etc. We've been through all that about mitosis and stuff to make sure the next cells also have those instructions. But that means that you're going to have something else that does the work. And the thing that does the work in the protein synthesis are RNA molecules. So the way it works is the DNA itself is a code. And it's a 3 base pair code, which means basically that 3 of the base pairs which we talked about being adenine, thymine, cytosine, and guanine, and the complement actually, the RNA is a little bit different, it's called uracil, but just serve the same function. But 3 of those makes codes for 1 amino acid. Okay. So since, like I said, the proteins are made up of chains of amino acids, you have a code that tells the cell basically what sequence of amino acids to put in there based upon the code, the 3 bases that are part of the DNA chain that it is reading. All right, we good so far? That makes sense? Okay. So matches the codon matches with a specific amino acids. That's what I just told you guys about. They're out there in the cytoplasm actually. So to start things out, an RNA molecule, a messenger RNA, comes into the nucleus, and it's a single strand. These are very similar to one of the strands of DNA. An RNA molecule is. And on a messenger RNA, it's got one particular part, so it's kind of like a strand here, like this, and we'll do diagrams later, but one particular part has just 3 bases on it. This could be A, C, and U. U takes the place of T, by the way. It's got other bases on there. It's a long strand, you know, a couple 10, 50,000 bases in length, but there's just one special three there that are out there that basically is the identity for that messenger RNA. And when it's time to make a protein, there's enzymes that establish that. They go into the DNA, they open up the DNA in a certain spot, they take out basically an area that is equal to one protein. That's called a gene. That's where the term gene comes from. So one gene makes one protein. So they'll go in there and open it up, and then these messenger RNAs will come into and match up with the portion of the DNA that matches those. So A goes to T, C goes to G, and U goes to A on there. Remember the T, the uracil replaces the T on there. So the U is going to go with the A on the DNA molecule. All right? And from there, when it fills in that DNA molecule all the way for one gene, and hits the end of that, the messenger RNAs all link up together. So now you've got, like I said, with 50,000 of these things all connected together in one long chain of messenger RNA. All right? That then is going to detach from the DNA and swim basically out into the nucleus, go through the nucleopores and the nuclear membrane, and go out in the nucleus. And it is going to, we're right here by now, by the way, it's going to go out and connect with the ribosome. So that actually, by the way, when it comes to RNA, the one RNA that doesn't really actively do anything is the ribosomal RNA. That's just what a ribosome is mainly made out of, is ribosomal RNA. Okay. And so it goes there and it connects in to that ribosome and kind of holds it in place, and it basically reads it like a chain. It kind of goes through it like a line through a donut or something. And it goes in there, and then the transfer RNAs start to do their job. So there are transfer RNAs in the nucleus, in the cytoplasm, transfer RNAs out in the cytoplasm already that are connected to amino acids, okay, specific amino acids. And those amino acids are specific for that 3-letter code that the transfer RNA will pick up. Now think about this. The messenger comes in and reads it. It gives what's called the anticodon to the code that's on the DNA. So if it's T, then the messenger RNA is going to be A. If it's G, then the messenger RNA is going to be C. Okay? And then when it goes out in the nucleus to the ribosome, it comes off there, the RNA, the transfer RNA is going to actually match up opposites. So technically, the transfer RNA and the DNA, they have the same codon sequences. Does that make sense? I'll do a diagram later that shows you that. But so you've got a code still. So you have these transfer RNA molecules. They have a particular 3-base codon that matches up. This is called an anticodon because it's the anti to the one that's in the DNA. So technically, like I said, the transfer RNA is actually a codon. So those 3 bases. Part of it's on there. On the other side of that tRNA molecule is the amino acid. And so they all line up on that ribosome strand, on that RNA strand, excuse me. And then they will, once the whole RNA is filled up with these tRNAs with amino acids on them, then the amino acids themselves will link together and peel off from there, and now you have a protein. You have an active protein. You've got a strand of amino acids in the right order, etc. The complete strand of what? There, group 1. Strand of what? Genetic strand. You guys go with that? Yeah? Yeah. Sorry. Protein is the answer there. Now, group 6, you put enzyme. Enzymes and proteins. I'm going to give it to you. I'm giving it to you because an enzyme is a protein. So, protein is the complete protein, actually. I should say they're not proteins. Complete protein detaches from the messenger RNA strand. That make sense? Okay, we're going to go a lot more details, show some diagrams, some videos, and stuff like this. I know it's complicated. Maybe we'll do a little acting out thing like we did with mitosis, meiosis.
Yes,
you guys
that
want to
was
do?
helpful.
Was that fun? Did it? Okay, good. So we'll continue on with that next time.